Two poor guys. Because two is better than one. We begin with a particular characteristic of DNA. It is a negatively charged molecule. We can detect it using a nanopore by applying a positive voltage on the other side to pull it through. As the DNA passes through the pore, it causes an impedance in the ionic current, which can be measured. But variability in the DNA's speed makes it difficult to examine features in detail. Compensating using gels or proteins perched on top of the pores helps, but error rates are still too high for commercial use. TPG's first invention addresses the DNA motion problem by using two nanopores instead of one, each with its own electrode. As before, DNA is pulled towards the first electrode, but now, a second electrode is amplified, pulling in the opposite direction. Because the DNA will move towards the stronger voltage, its motion can be finely controlled by adjusting each independently. As the voltages reach parity, the DNA slows. This model allows for precise detection of features of the molecule, or things attached to it. It's important to note that detecting the DNA is not the same as sequencing it. We can only measure current impedance from all the material inside the pore at a given time. If multiple nucleotides are inside, we cannot discriminate between them using current impedance measurements. But this is not a concern since we're not attempting to sequence DNA using this method. The two-pore method is used only to control the DNA's motion. Individual nucleotide detection can be done using auxiliary sensors within the two-pore architecture. Here, we show nanowires with a gap between them. As single-stranded DNA passes between the wires, a circuit is completed, and the nucleotide is identified. This method of detecting individual DNA sequences is called direct sequencing. There are a number of experimental technologies that have been shown to detect individual nucleotides, but there's never been a practical use for them without the ability to control the DNA's motion. So, they've remained solely experimental projects in labs. TPG does not make this technology, but is able to partner with those that do. In the meantime, there are more immediate uses of the impedance values from the nanopores that don't require auxiliary sensors. Namely, the pores can detect larger structures, including viral and bacterial proteins, antibodies, sugars, and other biomarkers of interest. More information will be made available soon. You can follow us on Facebook as well.